haven't met me or know me yet, apparently from the last class, it turns out that some of you were not aware that Monday was Walren's last lecture for the semester. But I will be taking over for Unit 4, so I'll be doing the next 12 lectures until the exam, and then you're going to have Dr. Tesfaye for reproduction. And I just wanted to take a minute too and talk about logistics, because I know as someone who used to get very stressed and confused about team taught classes, it could be a big deal to have to transition from someone that you've had for 3 units and you've gotten used to, and then suddenly here I am and you don't know what's going on. So I just wanted to reassure you that it's going to be fairly similar. I don't teach exactly the same way, but I'll be teaching the same things. I'm still going to stick to using outline boards because I think they're a great study tool. Not to mention, they're really good for keeping me on track of what I'm supposed to be talking about. And I'm going to draw. And I say this is kind of a good thing and kind of a bad thing, because the feedback I've gotten is that my handwriting's really nice and you can read it. Nobody really says anything about my drawings, and I think there's a reason. But we have PowerPoints, so everything I draw out, I will try to also show you in PowerPoint form so that we're clear on where we're at with it. Okay. As far as the exams are concerned, they're going to be the same format, 32 multiple choice questions and 3 fill in the blanks. I'll try to write a few of my own, and you'll be lucky if I do get that many, because they'll probably be easier, just because I don't write test exams as well as Dr. Walren does. That takes a lot of skill. But most of the questions will probably be from his backbone. I'll pull for questions from him and just update him for what I talk about. So you'll see pretty much the same thing even on the exams. Okay. I can't promise anything for Dr. Tesfaye. I'm not sure how he's going to teach, but you've got another 3 weeks before you have to deal with that. Do you guys have any logistical questions? Yeah. The house mic? Okay. How about that? Okay. So we're going to start with respiration. We're actually going to do 3 units, or 3 organ systems in this unit. We're going to do 4 lectures on respiration, 4 on renal, and then 4 on digestion. It's kind of nice because we're really going to take a deep look at the organ systems and how they work. And so you'll be able to tell full stories now. You could start putting together all the stuff that you've learned in the previous units into these organ systems. So the first one's going to be respiration, and this is tying in very nicely with cardio that you guys are going to have to take a test on tonight. Sorry. But we talked a lot about how the heart is just moving the blood around the body. It's taking it deoxygenated from the tissues, bringing it back to the right side, sending it off to the lungs to pick up more oxygen, and then sending it back to the left side of the heart and then to the body. So now we're just going to look at it from the lung perspective of how we're picking up that oxygen and how we can modulate the process. So the most important functions of the respiratory system are going to be gas exchange, oxygen primarily. That's what we've talked about already. This is what we're going to be picking up from the air around us. We're going to bring it into our lungs, and then we're going to move it across to the pulmonary capillaries so that we can get it to the heart and to the tissue. We don't talk about metabolism in this class, which is probably a good thing. It's a little complicated. But we will tell you that oxygen is what we're using to make energy. That's how we're going to make our ATP. And in the process of doing that, we're going to create CO2 as a waste byproduct. So we need to be able to get rid of that. CO2 is going to go the opposite direction. It's going to be coming back from the body in that deoxygenated blood. It's going to pass over into the alveoli, and then we exhale it. And then the third most important function is going to be the regulation of pH. So we keep our body's pH within a range of 7.35 to 7.45. That's a little higher than neutral, but it's very, very important that we keep it that way, because that's what our physiological systems need to be able to work successfully. We have a lot of enzymes that will not function properly if we get too far out of this range. So anyone who's been in a clinical situation, either working there or been in the hospital yourself, you've probably at some point or another heard them talk about acidemia or alkalemia. This is when you're too acidic or you're too basic, and it can be a medical emergency. It can actually be deadly. So you need to make sure that we're keeping this in that range, and we'll use the lung system to buffer that. Now, we're not going to get into the details of that today. I just wanted to bring up the functions, but we'll talk about this probably Monday. Today we're going to focus more on just the basic anatomy of the system. So we're going to talk about the conducting zone, which means tubes. Okay, we've got to get the air in somehow, and we're going to move it through these tubes. That's going to be the larynx, and then the trachea, down to bronchi that are going to separate to go to the different lungs. And then it's going to branch out into all of these bronchioles. After that, it moves into alveoli. But we consider this to be respiratory zone because this is the functional unit of the lung. This is where we can actually do gas exchange. So we'll move the oxygen in and move the CO2 out here. But the lungs don't work by themselves. In the thoracic cavity, we have other anatomical features that will help us to be able to breathe in and out. One we've seen before. We've seen mesothelium when we talked about heart. We called it pericardium then. But it's the same principle. It's this fluid-filled balloon that your organs grow into, and then you'll have one side, the visceral side, that's going to be directly connected to the organ, and one that's going to be connected to body wall. Between it, we have fluid that allows it to move nicely without friction. So that's going to be really helpful when we're expanding and then relaxing our lungs. And then finally, we're going to have diaphragm. So diaphragm is actually a skeletal muscle that separates your thoracic cavity and your abdominal cavity. And it's dome-shaped. And we can contract it and flatten it and then relax it again to change volume and affect pressure. And that's going to affect our breathing. So we're going to look at how we do that. But we know that since it's skeletal muscle, we already know the story. You guys have done this. You know the idea of having a lower motor neuron going to a muscle fiber. We know the neurotransmitters and the receptors and everything else that's happening. The only difference here is that where we used to talk about lower motor neurons in a very general term, now we're actually going to name it and say that this particular lower motor neuron going to your diaphragm is part of the phrenic nerve. Okay. Finally, we should be able to get into ventilation. That's the actual process of breathing in and out. We'll start with inspiration, which again is caused by contraction of your diaphragm and a change in volume. And we'll look at how that change in volume can cause that change in pressure that brings the air in. I didn't have room to put it on the board, but we may even get to expiration because it turns out I talk faster than I thought. So if we do, it's essentially just the opposite of what's written here, and I'll write it out on the board if we get there. Okay. So we'll start with anatomy of the conducting zone. So it's this anatomy of the respiratory tract. Really, this is starting in either your nose or your mouth. That's where the air first comes in, but we're going to show it starting at the trachea. Actually, we're going to go back one and show it at the larynx. Okay, so your larynx is part of your throat that has your vocal cords. So this is a vocal cord. Or more technically, a vocal fold. These can vibrate. So as air passes by them and they vibrate, you can make sounds. If you're like me, you're lucky to actually get the words out that you want. But some people are actually trained to be able to move these so precisely that they can hit notes on request. I have no hope of that. I'm not even going to try. I'm glad I can speak. But the larynx is not just a tube for getting air through. It's also important in our speech. From here, we're going to move into trachea. Trachea is just a fairly flexible tube. And that becomes a problem. Because when we start messing with volumes and changing pressures, we will create negative pressures in our lungs. That negative pressure will try to collapse the trachea if it doesn't have some sort of support. So we have, I used orange, we'll do red, these cartilage rings. That prevent this from collapsing. So you can actually feel that in your throat. If you're rubbing up and down, you can feel those cartilage rings. And in fact, if you've ever seen a movie that has a diner, like especially if it's from the 80s or 90s, I don't know why they were just obsessed with it then, but if they ever had a diner scene, you could almost guarantee someone was going to choke on a pie and another customer was going to stand up, pull out a pocket knife and grab a straw and go, I got this, and then just cut a hole in their trachea. That's what they're doing, is a tracheotomy. You're trying to get past where the food is stuck so that you can still have air come in and out, bypassing that. When you move past the trachea, you have to start splitting because you have a right and a left lung, so you need to be able to get air to both places. This is going to be right and left bronchi. Besides just being a passage to each of those lungs, bronchi become really important because they have smooth muscle. So we haven't talked a lot about smooth muscle. We talked about it with arterial modulation, okay? But all smooth muscle is going to be under autonomic control. So this means that we can control this and we can modulate things. If we're talking about smooth muscle around a tube, most likely we're talking about changing radius because we can contract or relax that muscle. So probably on Monday, we're going to talk about all the things that we can do to be able to modulate this bronchi and the clinical implications for it. For now, we're just looking at it as the tube to get the air through. From these bronchi, you're going to split off into hundreds of thousands of bronchioles. Really, when you think of lungs, if you ever see pictures of those pink squishy, or if they showed you people who smoked in school and they're black and stiff, those are really all of these bronchioles and then the alveoli that are off of them. That's what makes up your lungs. So if I were to draw alveoli in here now, I would just try to draw grape clusters, basically, kind of what it looks like. And I don't know what I drew there. That's like a impossible, what do you call those Escher thingies? We'll just do that. Voila, fixed it. Okay, so when we're talking about everything up to the bronchioles, whoa. Again, this is what we referred to as conducting zone, meaning that we're just moving that air through. We can't exchange gas here. The alveoli is important for that gas exchange. So if we take a closer look at the alveoli, we would see that the alveoli is a single epithelial layer thick. So these are epithelial cells. We know all about those too. We know that anytime we have epithelium, it's going to be nicely organized. It's going to be connected to one another. It's going to be oriented, right? So anytime you see epithelial, you know there's going to be a basal layer. And I will draw that in as tick marks. And then an apical, which is usually facing a lumen or an outside world. In this case, your lumen is where you get the air in. So this is apical surface. You're going to have a thin layer of fluid that lines every one of these alveoli. And that's going to become really important in the story of how we're going to open and close these alveoli. So just like epithelium everywhere else we've talked about, there's particular names, right? I know it's a pain to have to learn them, but once you learn them, those names tell you where you're at. Here in the alveolus, epithelium is referred to as pneumocytes, and you have two types. Type 1 is going to be gas exchange. I'm going to say all these black ones that don't have green in them, those are type 1. They're the most prominent, the one you're going to see the most of. But we also have type 2. They're going to be these green-filled ones. They're not as common, but that doesn't mean they're not important, because these type 2 pneumocytes are going to secrete surfactant. One of the surfactants it secretes is called phosphatidylcholine. Okay. That's a pain of a word, right? It's really big, it's really long, who cares? But it's actually important because it tells us a lot just by reading it. It sounds very similar to words we've talked about before. It's got a phospho, it's got this choline. So hopefully this is making you think of the phospholipids we've talked about. And it is very similar. It is amphipathic. Amphipathic. Meaning it has both the hydrophilic and the hydrophobic side. So it's going to have a charged head group, and it'll have hydrophobic tails. These are detergents. They're really good at being able to get in between water molecules and separating that surface tension. Because you remember, hopefully, although this was Unit 1, that we have water molecules that are an oxygen and two hydrogens, and that the oxygen has a partial negative, and the hydrogens have partial positive dipoles. And that means they can interact with other water molecules. So now you can have this hydrogen interacting with a partial negative of this oxygen, and on and on. So that was what created that surface tension. We have surface tension here in the water layer that surrounds the alveoli, or is, I should say, on the inside of this alveoli. And because these alveoli are very, very small, and they're like little balloons that can stretch out and then come back again, this water can be a real problem. Because not only is it creating a surface tension with the water molecules next to it, if you get those two layers of water close enough, they're going to start interacting. And then when you want to try to open that alveoli again, you have to break apart that surface tension to do it. That's where the surfactant is going to come in. This detergent will help reduce your surface tension so that you can separate the two sides of the alveolus and open it up. The other main component here is going to be those pulmonary capillaries. We can bring air in, but we still have to be able to move it across into our blood. So we have pulmonary capillaries that are going to wind around every alveoli. They're in very close contact. And a capillary, I want to do that in orange, is just one endothelial cell thick. To allow for that diffusion. So this is coming in from a pulmonary artery. Remember, this left the right side of the heart and got shipped out to the lungs. And it's going to bring with it erythrocytes that are deoxygenated. So we have low O2. This side is actually going to be going to the pulmonary vein. and going back to the left side of the heart. So it's bringing that oxygenated blood back so you can distribute it to the body. Because you have low O2, you start moving across the alveoli in the pulmonary capillary, but you have high in the alveolus. If you've just taken a breath in, there's a gradient. And gases will move down their gradient just like ions and water and everything else we've talked about. So with higher O2 in your alveolus, move across a type 1 pneumocyte, pass through the endothelium, and then bind to a hemoglobin molecule on your erythrocyte. We've talked about diffusing, moving things across before. We've talked about capillaries and lymph. But in that case, we talked about stuff just being pushed out into tissue that's pretty fixed. Now we're talking about trying to move gases across 2 layers and then attach to a moving target. These erythrocytes are moving past here in like less than a second. So you don't have a lot of time to be able to grab that oxygen. That means you need to move it across as quickly as possible. And that's why you only have one epithelial layer in the alveolus and one in the endothelium. That's also why this space between the two is really small, maybe about 5 micrometers. I don't care about the number, don't memorize the number, that's just for graphic purposes. Okay. So again, this is clinical implications. If we cannot get the oxygen across fast enough, then we can't load it, and our erythrocytes go to try to deliver oxygen to tissues, and there's nothing to drop. So you're in trouble. This can happen whenever you make this bigger, like the diffusion pathway is longer. So you can imagine if you had more fluid in your alveoli than you should. Say you had pneumonia, which often leads to a buildup of fluid in that alveoli. That's all that extra fluid that gas has to move through before it can cross over. It's going to take it a lot longer. You can also have diseases where you'll get scarring. The tissue gets thicker and it gets harder and it's not as easy to move across. All of that can slow down your gas diffusion and means it's much, much harder for your erythrocytes to pick up the fresh oxygen. Okay. People with like COVID pneumonia or any pneumonia in a hospital, one of the first things they tend to do is put them on high flow oxygen. It's a high concentration, because if we can increase the amount of O2, we increase the gradient and more oxygen will start to move across. So there's a better chance that the erythrocyte can pick it up. If that's not working, they may also start using more pressure, literally trying to force that oxygen across the tissue so that it gets to the erythrocytes. Okay? So the other thing that we have to worry about is not just moving O2 in, but again, moving CO2 out. So we've got this waste product we need to get rid of. So here, we're going to do high CO2 levels coming back, and we have low CO2 in the alveolus itself. So the gradient is to move from the erythrocytes across the endothelium into the epithelium, and then move, exhale it out, I'll say. So I'm going to draw this again. Here's an erythrocyte. It's got a CO2 on it. That CO2 is going to move across and out. Okay. Questions up to there?
So with a disease like pneumonia, eventually, like lungs will clear themselves out of fluid. What do you do if you have scars? Is that something you can fix, or?
The question is, what do you do if you have scarring from something? Like pneumonia, you can recover from, but sometimes it'll still do damage that's lifelong. Your pneumocytes are not particularly good at regenerating, so a lot of that scarring is going to cause problems in the long term. Keep in mind, we have a lot of alveoli. So as long as it's not too drastic of a scarring process, you'll still probably be fine. And I'm sure they probably have medications to help with that as well. Yeah.
Does the exchange of carbon dioxide and oxygen happen on the same erythrocyte, or is it like one erythrocyte does the oxygen and the other one does the carbon dioxide?
So are we exchanging CO2 and O2 on different erythrocytes? No, you're pretty much doing them at the same. So you want to dump that CO2 off, and then you want to pick up new oxygen. We're going to talk about CO2 in more detail, and we're going to find out that the CO2 on the hemoglobin is rare. Most of our CO2 that we're getting rid of is actually coming in the form of bicarbonate. So we have a lot of CO2 on this side, so we do have a gradient, but it's not all actually connected to the erythrocyte. I was just fibbing to make it simple for now. Yeah. Anything else? All right, I want to show you some pictures just to make sure you can visualize this. Okay, so we talked about the larynx, which is where we have our vocal cords, the trachea with its cartilage rings to help keep it from collapsing, then splitting off into the right and left lungs with the bronchi, and then the bronchioles going to alveoli, so our little grape clusters. I don't like that picture, I've got a better one. But first I just wanted to talk about how many of these ducting systems we have. This is a little more detailed than we're teaching in this class. Don't worry about the different types of bronchioles that are listed. We just want you to know bronchioles. So if you go from one trachea to two bronchi, then you get down to the bottom bronchioles, you've got 500,000. Once you actually get down to your great cluster alveoli, it's 8 million. Okay, so what I've always heard is if you were to take all these alveoli and lay them out, they would be the size of a tennis court. So that's an enormous amount of surface area that we can do this gas diffusion across. Here's a really great picture of how those capillaries are interacting with the alveoli. So we have these great clusters, and then we have all of the little capillaries really wound closely to them. We're coming in deoxygenated from the pulmonary artery, and then as it picks up oxygen, it's painted red here, and then it's going to join back together to be the pulmonary vein and go back to the heart. And then I love this. This is a cross section so that you can actually see the alveoli and two of the capillaries. You can also see an erythrocyte, so it really gives you a good view of how tight those are. Those erythrocytes have to go through capillaries single file. Okay. All of the pink here is meant to be type 1 pneumocytes that you're doing the gas diffusion. This blue alien blobby thing is meant to be a type 2 pneumocyte. So that's what's going to be important for surfactant that breaks up the surface tension. And then you have to move the gas across the epithelium into the endothelium and then onto an erythrocyte.
What's the purpose of having fluid on the outside if we're just constantly break it up?
So what's the purpose of having that liquid on the inside of the alveolus?
Yeah.
I'm sure there's a lot that I don't even know to tell you, but I know for one, all of your gases, your oxygen in your body, like your blood, in your cells, it's all in liquid. So we have to actually get it into solution and then move it across. Okay. So that's our transition from air into solution.
Okay.
Anything else? All right, we'll talk about the rest of the thoracic cavity. There we go. Sorry. I love these boards, but they did not have short people in mind. All right, so I'm going to redraw the structure of the conducting in respiratory zone, but in a lot less detail. So I'm just going to have a tube coming down and splitting off, and splitting and splitting. I think I'm getting better. All right, so if we were to draw in a shadow of our lungs here, oh, I shouldn't have done that quite so big. We're going to make them short lungs, like me. The first thing we want to talk about is the pleura. Again, that's the mesothelial layer. We've got the parietal and the visceral. Visceral is the one that's going to be directly on the organ itself. So we'll do that as blue. Usually these visceral layers are so intertwined in an organ that it's really hard to even see them. So that's your visceral layer. But of course we know this is a closed sac. So you have your outer layer, which is the parietal layer, and this is going to be connecting to the thoracic wall. And again, visceral's on the organ. And together, that's your pleural sac. Like all good mesothelium, you're going to have a thin layer of fluid in here. That fluid is really important for being able to move these two layers without friction. So again, as you inflate and deflate your lungs, you need to be able to have these two layers move without having too much friction. So you've got this nice smooth fluid that allows that to slide past one another. But just like we talked about with pericardium on the heart, you want to keep that to a minimum. It really is just a very thin layer of fluid. You start putting too much fluid in here and you're going to get a problem. Okay, so if you build up too much fluid, then you're going to have a hard time being able to inflate your lungs against that pressure. So here's a mantra for this unit 4. The pleural sac will always be negative pressure relative to the lungs and atmosphere. There's some other really cool clinical implications for this, like pneumothorax, and we'll talk about that. We'll walk through a pneumothorax and what's happening. So our other player here is going to be our diaphragm. That dome-shaped skeletal muscle. Oops. So remember that skeletal muscle is going to be innervated by lower motor neurons. And hopefully you guys remember where lower motor neurons are coming from. I will draw in, I'll draw it here. Here is a cross section of your spinal cord with your favorite butterfly. We remember that this is dorsal side and this is ventral. Motor will always be on the ventral side. Efferents are coming out of the ventral. So you're going to have lower motor neuron cell bodies in this ventral horn. This is a cervical spinal cord. I'm going to label that. Not because I care that you know it's specifically the cervical section, but just so that we're oriented. Cervical means neck, okay? So the lower motor neurons that are feeding your diaphragm are actually coming out of your neck area. And they're going to go from the ventral horn, out the ventral root, and we're going to join together into a spinal nerve. So those spinal nerves that we have are always going to be carrying the sensory afferents coming in and the motor efferents going out. So you're going to have a lot of lower motor neurons, more than I can draw in this tiny picture I made. But we'll just show 3 coming down here to innervate. But they're all being carried together as a nerve. The phrenic nerve. I really just want to take a second and redraw that because that's awful. We're going to stick this here and say here is our spinal cord. So we've got all of these lower motor neurons that feed out and become part of the phrenic nerve. Okay. When these phrenic nerves innervate the diaphragm, they're going to do exactly what we've talked about before. They're going to release acetylcholine, and that acetylcholine is going to bind to acetylcholine nicotinic receptors on the skeletal muscle. That causes the influx of sodium, which causes an action potential, travels down the T-tubule. That's going to open the DHP, like it's going to pull the foot out of RYR's neck and allow that calcium to come out of the sarcoplasmic reticulum. It's going to bind to troponin C, move tropomyosin, your heads come up, grab the actin filaments, and power stroke. So this is going to have everything that we've seen with the Z lines and the sarcomeres, your actin thin filaments, your myosin between. Okay, same story. And that may seem surprising when I tell you that this is what's causing you to be able to breathe in. Because most people think breathing is automatic, and you're right, it is. But we can actually control it through a skeletal muscle. We can choose to take a breath in when we want to. We could choose not to breathe, at least for a while. But if you stop breathing long enough, you'll have sensors that will go off and they will try to force you to breathe. Okay, you'll send signals through an autonomic system direct to this muscle, trying to make it contract even if you don't want to. If you can actually withstand that somehow, you'll probably pass out from lack of oxygen, and then it's just going to kick in anyway. So regardless, when your brain wants you to breathe, you're going to breathe. Doesn't matter if you're underwater, unfortunately. That's how you end up with water in your lungs when you drown. Okay, so how is this actually relating to the idea of being able to breathe? We have this muscle, it's domed. So it's taking up quite a bit of space in our thoracic cavity. When we contract it and we shorten these sarcomeres, we're going to cause this to flatten. So this is a contracted diaphragm. For inspiration, which just is a fancy word for breathing in, that contraction of the diaphragm is what allows the air to move in. So this is going to be caused by diaphragm contraction. And to understand that, we have to look at some chemistry. Sorry, I know it's no fun, but it's fairly simple. This is just Boyle's law. Which says that if one container has a pressure and a volume, and that container is connected to a second container, then the pressure times the volume of the two containers are going to equal. Now, this is only for connected containers. But when we're talking about the thorax, it is connected to the atmosphere. There's no sphincter. There's no way to block the air from the atmosphere to our lungs. So if we look at this container as being atmospheric, pressure is constant at any moment. You know, pressure could change depending on where you go altitude-wise, but all in this room, we're all experiencing the same pressure. Volume's the same, probably like infinite. But when we look at our thoracic cavity, we can start manipulating this. We already saw one way. We flattened this diaphragm by contracting this muscle, and we've increased our volume. If this has to stay the same, and this increases, then the pressure has to drop to make up for it. Hopefully that's intuitive too. We kind of understand that if we have the same amount of air in a bigger volume, that not as much of it is going to hit the walls and cause that pressure. Okay. So for a moment, as we drop that diaphragm muscle and contract it, we're going to drop the pressure. It's not going to last long because we're directly connected to the atmosphere. So as soon as we do that, we essentially create a vacuum that's going to suck the air in from the atmosphere and into our lungs. Okay, so this is, we're going to say, let's say P1, not I, is greater than P2. And again, you can look at this as a gradient. We haven't really talked about pressure gradients before, but it follows the same principle. If pressure's higher on one side, it's going to move to where it's lower. So this goes down its gradient. This is going to create negative pressure in the lungs. That you can think of as a vacuum. You don't so much inhale as you just suck the air out of the atmosphere. I'm still early. How'd I do this? Any questions up to here? Okay. We'll go ahead and start talking about expiration then, because I'm still talking too fast. Expiration, like I said, is just the opposite of inspiration. This is breathing out. I know why I forgot something. Let's put expiration on hold and come back here for just a moment. We need to point out that this process of inspiration is active. We're using skeletal muscle. We're using ATP to be able to release the heads, reset them, and keep cranking them down. So we're using a lot of energy to be able to change this volume and create this difference in pressure and bring the air in. So we consider inspiration to be an active process. Expiration, on the other hand, is considered passive. We basically just stop contracting. The volume is going to reverse, and then the pressure will do the same. So expiration, breathing out, relaxing our diaphragm. So now if we look at our Boyle's Law equation, when we relax that muscle and it goes back up to being fully domed, we've reduced that volume. You reduce the volume, you have to make up for it by increasing pressure. Again, it's momentary, because now you've got a gradient where the pressure in your lungs is slightly higher than the atmosphere, so it's going to push out. Or you could say the atmosphere sucks it out. But we do have a few additional details to expiration. Okay, so it's not just a matter of relaxing the diaphragm. We're also going to utilize something called elastic recoil. This is the idea that when we were able to breathe in and we were able to expand the alveoli, we're going to store some energy in that process. And when we snap back, when we have a recoil so that that balloon starts to close again, that passive energy that was stored will help push the air out into the atmosphere. And I'm going to leave it there because I don't have time to get into that. So you guys could do a little more studying for the exam tonight. Good luck on the exam, and I'll see you guys tomorrow.